we want to live longer, but also we want to live healthier. So we've done a great job in medicine in keeping people alive longer. Other aspect to living longer is living healthy. And this is healthspan. And right now in the US, we're looking to over a decade of living in ability independence. Part of what we need to do is not only live longer, but narrow that giant gap that we've been creating. Let's live happily, and your families live happily. How do I think we can do this? Regenerative medicine. It's got to be part of the equation. No questions in my mind about this. I'm Anthony Oliva. I'm Chief Scientific Officer for American Cell Technology. This company is a autologous cell banking company, if you're interested. And we're one of the largest cell banking companies in the US. I just want to talk to you a little bit about general things when it comes to MSCs. Okay? These are cells that can be culture expanded to high purity. You can get them from umbilical cord blood. Most of what is done at American Cell Technology is donors getting it from adipose tissue, so you can strategically remove a little bit of fat. It comes to the company, and they can be culture expanded to high purity and then biobanked for later use. And people also do it with bone marrow, but by far, adipose tissue is the way to go, especially as an adult. If you want to use MSCs for yourself or your patients, there are things I want you to be aware of, because as someone brought it up earlier, this field is a little bit like the wild, wild west sometimes. It's gotten much better, but in the early days it was. But still, things to look out for. You first and foremost, you want a company that is making stuff under using good manufacturing practices, okay? If they're not using GMP to make the stuff, you should find someone else that is, okay? Because that directly gets down to safety. You want a safe, reliable product. You want to make sure you're not introducing some adventitious virus to a patient. The cells need to be MSCs. There's lots of ways to characterize them. That can be done by flow. They also have the ability to differentiate into multiple tissues. And then one of the other key aspects of mesenchymal stem cells is that they are physiologically responsive. So you can give them to a patient, and they can actually tailor their responses to the environment that they land in. They're chemo-attracted. They're home to sites of inflammation and damage. When they get there, they can change the response to help initiate the repair mechanisms that are actually required. And so, and I bring this up in the context of, well, do I use exosomes? Do I just use the MSCs directly? And the answer to that is, depends on what you're treating. And maybe a combination of both. Exosomes are not, exosomes, they have to be tuned to what they needed to do ahead of time. The MSCs directly don't have to do that. They're called stem cells, but it's a bit of a misnomer. But suffice it to say that these cells, you can't culture them indefinitely. You can culture them for a limited number of replications, and then they start to senesce. They die. And usually that's in the ballpark of somewhere between 3 and 6 passages, if you're looking at passaging. So what does that mean? Well, if you have a company that says, well, we can make 100 trillion cells from your adipose tissue donation, and we can grow it out for 10, 20 passages, yeah, maybe they can do that. But what I will tell you that you will be getting is not very good MSCs. When they start to get out of this linear growth phase, they're actually going to transition from being a pro-regenerative phenotype to a pro-inflammatory phenotype. And then when you start getting the accumulation of senescent cells in there, you're poisoning the whole system. So, so these are just practical things to be aware of, and questions if you do, have a company that you're considering. Those are things to ask them. We hear a lot of benefits of MSCs and why, these cells do everything. And there's a lot of trials out there that are done, a lot of equivocal results coming out of it. But when you look at the MSCs, these cells, they're natural residents of vessels. They hang out in the periphery, and one of their main functions is to help repair and regenerate your vasculature. As you get older, vasculature starts to become dysfunctional. Give cells back, you can make it better. The common theme of the vascular dysfunction and sort of broadly in aging-related indications. So I'm a neuroscientist by training. One of the things I think this can help with is any type of neurodegenerative disorder. In terms of frailty and cognitive frailty, to me, Alzheimer's disease is really like a great example of something you can potentially treat with these cells. That is the definition of unsuccessful aging of the brain. In something like Alzheimer's disease, one of the key things that goes on is the dysfunction of the vasculature, and maybe the lymphatic system as well, most likely. So that was whole brain hippocampus, okay? So for those of you who don't remember what the hippocampus is, it's that deep brain structure. In the cortex, this is where you form new memories. And the hippocampus is particularly important in the context of Alzheimer's for two reasons. It's one of the first structures that really goes. It's also one of the two places in your brain where you're constantly regenerating cells. The other place is the olfactory bulb. So this has constant turnover. You're turning over new neurons in this every 2 weeks. Autologous versus allogeneic. Both can be used safely, but there are some advantages with the autologous, in my opinion, and part of the reason why I went from an allogeneic to autologous. When you're trying to treat large indications like AD, where you need hundreds of millions of cells, you can't find enough donors to make an allogeneic product to meet the market. It's not possible. You can't, if you have millions of people with AD and you can only get a few hundred to 1,000 doses out of a single donor, you can't treat that entire market. The other real advantage to the autologous MSCs is that they are tissue type matched. What does this mean? Well, they now have the ability to actually actively participate in the regenerative or repair responses. Okay. One huge benefit versus the allogeneic. They both do the same things to a point. Allogeneic acts like a ringmaster. Okay? You give them, they hang around for a while, they tell all the other cells or whatever it is, kick in the system, here's where you go, and then the exit stage, and they're gone. The other real advantage to the autologous MSCs is that they are tissue type matched. What does this mean? Well, they now have the ability to actually actively participate in the regenerative or repair responses. The central nervous system on its own just does not spontaneously regenerate because you gave it a monoclonal antibody. This is a situation where if we're actually going to treat this and prevent it, a regenerative medicine approach has to be part of the equation. This is the foundation of what I would consider is probably going to be needed is a true combinatorial therapy approach to keeping these patients alive and healthy and actually cognitively working.